So kia ora and hello everyone. Welcome to today's webinar, Multiplication and Division, Monitoring Cell Proliferation by Dye Dilution Analysis. It's my absolute pleasure to introduce our webinar speakers today. We've got Kathy Muirhead and Paul Wallace. Dr. Kathy Muirhead is Chief Operating Officer and co-founder of CYTO Inc., which is a biomedical consulting group. Over the course of her distinguished career, she has served as Assistant Professor of Pathology at the University of Rochester, Director of the first Flow Cytometry Core at SmithKline Beckman R&D, and Senior Vice President of Research and Business Development at Synaxis Incorporated. Kathy has played key roles within ISAC, including serving as counselor and editorial contributor to Cytometry Part A. She has also helped shape clinical practice through her work on CLSI subcommittees and is a co-founder of the long-running and flow cytometry rite of passage annual courses and applications of cytometry, which is an educational series that's now approaching its 50th anniversary. She currently serves as on the ISAC governance committee, where when I was chair of that committee, she was absolutely crucial to the success of the things that we did then, and I'm sure that continues now. And joining her is Dr. Paul Wallace, legend, Professor Emeritus at Roswell Park Comprehensive Cancer Centre, where he served as Director of Flow and Image Cytometry Department from 2003 to 2021. Paul is currently the Educator-in-Chief and a past president of ISAC. He also serves as the Chief Scientific Officer of CYTO and remains actively involved with the International Clinical Cytometry Society. He's an Associate Editor of Clinical Cytometry B, and in 2018 was honoured with the Wallace H. Coulter Award for Lifetime Achievement in Clinical Cytometry. We will have time at the end for questions, and if you have any, please just pop them in the Q&A chat box. And without any further ado, I'd like to hand it over to Kathy and Paul, two flow cytometry and proliferation gurus, to hear today's webinar.
All right, so the topic today is multiplication and division, cell proliferation monitoring by dye dilution analysis. Cell proliferation is a topic of interest to cytometrists since since the very beginning of the field. And basically, proliferation represents increases in cell number due to cell growth and division, and decreases in cell number due to cell death or differentiation as they exit from the cycle. And with that, as a quick intro, Kathy, you want to take over the disclosures?
Yeah. Okay. I will point out that, yes, we are both employed by CYTO, which is a biomedical consultancy. And one of CYTO's clients is Phanos Technologies, Inc., which owns the PKH and CellView dyes, some of which you'll hear about because they're part of one class of proliferation dyes we'll be discussing today. In prior lives, Paul and I have both received either pre-commercial evaluation samples or complementary samples of various proliferation dyes that are sold by a variety of companies. And so we have had a chance to play with many of these, and we really appreciate their being willing to do that. But, you know, they've donated to us, so thank you. I don't think it's a conflict of interest, but it's at least something you should be aware of. And I guess you could carry on then, Paul.
You want to do the...
Sorry. Yeah, sorry. The agenda for the rest of today is basically to take a quick look at DNA cell cycle analysis, because that was really probably the first where proliferation analysis by flow cytometry started. Cells have to make DNA, synthesize DNA, and by looking at their DNA content status, we can say something about where cells are in the cycle, what proportion of a population is cycling, etc. So we'll briefly look at that, but then most of the rest of today will be focused on dye dilution, which provides a different way of assessing cell proliferation by assessing the extent of cell division in a population that's been labeled at time 0 and then followed after some kind of stimulus. And Paul will walk through the principles, the different types of dyes that are used for this kind of dye dilution analysis, and how we would analyze and report the extent of proliferation based on dye dilution. We'll each cover a couple of applications, and then I'll talk a little bit about the controls and troubleshooting for the most sort of typical FAQs where things can go wrong. And then it'll be your turn to ask questions.
Okay. So we're going to basically, I'm going to start off with a little bit on the cell cycle, as Kathy said, and then we'll move into a talk on dye dilution. We'll switch. Hopefully, we'll be able to switch in between, and Kathy will take over the latter part of that piece. I'd point out that part of this is from content that we have as articulate modules in CYTO, which was actually three different lessons. The very first lesson was more of an intro. The second lesson is really focused, or the second group of modules is really focused on cell cycle, and the third one is more on proliferation. So lesson 1 of the CYTO proliferation course reviews the cell cycle as a tightly regulated process by which normal cells grow and duplicate through DNA and divide to produce 2 daughter cells. A DNA histogram provides a nice snapshot of cell cycle status at a particular point in time. And with a perfect cytometer, the cell preparation method and saturating amounts of a DNA binding dye, an exponentially growing cell population will give a histogram just like this. Of course, that's not what we ever see. We could identify then quantifying percent of cells in S phase by simply putting analysis regions to define that region. If we had mixed a second aliquot, shown here in red, of normal cells, and I typically use quiescent lymphocytes, having a diploid content, we would also determine where that population fell, and that would be considered a normal or abnormal. In this case, these cells would be hyperdiploid. Lesson 2 of the CYTO proliferation course describes how modeling can be used to analyze the shape of real DNA histograms, where staining and measurement variability cause overlap, as you can see in here, of the early S phase cells with early G0, G1, and of late S phase cells with the G2M population. Lesson 2 of the proliferation course also describes how proliferation-associated antigens can be used to provide more detailed cell cycle analysis than is possible with DNA content alone. These include proteins with DNA synthesis, the pCNA, Ki67, the phospho-H3, the cyclins, etc. But I would warn you that fixation and permeabilization protocols for each target antigen should be followed closely, since they differ for cytoplasmic versus nuclear antigens, and are different in many, and also differ in many ways for the phenotype of interest. Diffusing fluorescent proteins to orthogonically express cell cycle regulatory proteins creates biosensors, these Fuji as an example, that are able to report real-time cell cycle status in living cells. So you can see the quiescent here could be red, and the ones that are going through S and G2M are in green. And that's really illustrated here, where in the inner cells that are proliferating are green, and you can see up here or actually this is BRDU, but they're right in here, and you can see the red ones are the ones that have fallen out of the cycle, and that's seen here. So the red are high in G0, G0, then they become positive for in G1, and then they lose their Fuji when they in RNS, and they pick up the green. So that's really what you're seeing here, and you can see that that's true. Here's the pCNA, and those are all taking up the pCNA, and here's the BRDU, where they're taking up the BRDU.
Everyone, so sorry, it looks like Paul's just frozen for a minute. What we're going to do is we're going to ask him to rejoin. Here he comes. and hopefully we can carry on from there.
Okay, apologize. So basically, I think I've gone a little bit ahead, but here as I was trying to figure what's going on. But just this is this, as I mentioned, and that's why I got Kylie kicked me off. That this is our hero Kylie, and basically this is being a histogram doesn't tell us anything about how rapidly our cells are progressing through different cell cycles, but metabolic labeling with thymidine analogs such as BRDU or the clicket reagent EDU provides improved resolution of S phase for standard cell cycle analysis. So here T0, you can see that all the cells have taken up the, in this case, it's the clicket EDU. These are HL60 cells that were pulsed for an hour with EDTU, and then returned to culture and sampled at 2-hour intervals, and then counterstained with DAPI, as you can see down here. The gating on EDU positive cells, which is here, shows that really all the cells that are in S phase have taken up, and there's really none left over in G0, G1, or in G2M. If you look then as you move down through time, so this is 2 hours and 4 hours, you can see that a lot of them now are accumulating in G2M, and there begin to be some that appear in the G0, G1 phase. The percentage of EDU stayed constant throughout the experiment, indicating that the labeled cells and the unlabeled cells were dividing at the same rate. And that therefore EDU incorporation hadn't altered cell cycle kinetics. In vivo labeling using long-term administration of thymidine analysis is also something that is used by adding it to drinking water to follow cell populations in animals. One thing I would point out is that not only is the numbers, while the numbers staying the same, you can actually see the cells moving through the cycle, because as they divide here, the intensity is about half as they divide and move into G0, G1. And in fact, you can see that here they're all in S phase here, and by 24 hours, they're all back in S phase. So our cycling time or our doubling time for this particular cell is 24 hours. So I'm going to move now into the dilution. We'll talk a little bit about principles. So time-lapse imaging of Fuji reporters can tell us how many cycles a given cell has gone through, but really it's impractical to follow large numbers of cells this way. High concentrations of thymidine analogs cause cell toxicity, so they can only be used to follow 1, maybe 2 rounds of cell division before their intensity becomes indistinguishable from unstained cells. Dilution proliferation, all assays address both of these limitations. A practical requirement of proliferation tracking dyes is that they are stable and non-toxic when incorporated at high levels. They should distribute approximately equally between daughter cells at each division, and their intensity dilutes out constantly from generation to generation. The consequences of this differential is shown here, where one cell, that's this one cell here, in 10, which is basically the parental generation, responds with stimulus by proliferating with half of the dye going to each daughter cells and on down. Responder cells outgrow non-responder cells after 4 rounds of cell division. So by here, you can see that the responder cells are outnumbering the 64%, are outnumbering the number of non-responding cells. And in the 5th generation, 1 in 32 are as bright as the cells that haven't undergone any division if intensity is divided in each division. The advantages are that proliferation dyes can be combined with antibodies, antigen-specific multimers, or other probes to quantify subsets of interest within a complex population and determine the extent of cell division without having to physically isolate them for counting. Parental and or daughter cells that respond differently to agents that stimulate or inhibit cell division can be physically sorted for further functional and genetic characterization by other methods. Some, but not all, the dyes used for general cell tracking can also be used to monitor cell division. To be useful for proliferation monitoring by dilution, a tracking dye should exhibit stable cell association. It should also have no effect on viability, growth rate, or other functions when cells are labeled with starting intensities high enough or bright enough to follow the desired number of cell divisions. In addition, it should be verified to exhibit a linear correlation between fold decrease in intensity, dilution that is, and fold increase in cell count in a simple system where a single cell type is growing logarithmically. Ideally, fluorescence should be halved at each cell division. However, even in situations where other factors such as protein turnover or dye metabolism result in intensity decreases greater than twofold, dye dilution can still be used to estimate the extent of cell growth so long as rate of dye loss due to such factors is constant across generations. Commercially available cell tracking dyes vary widely in their chemistries and fluorescence properties, but the great majority fall into one of two classes based on their mechanism of cell labeling. Protein dyes, shown here, typified by CFSE, are amino-reactive dyes that form stable covalent bonds with cell proteins. Membrane dyes, on the right, typified by PKH26, are highly lipophilic dyes that intercalate stably but non-covalently into the bulk lipid phase of cell membranes. Both classes of dyes come in a wide range of colors, and each class has its own advantages and limitations, which we'll discuss. The key to successful use is therefore understanding the differences and how to optimize staining and analysis strategies for the experimental system, and of course, your question. One important difference between the dye classes is how soon after staining a dye dilution can be initiated. Quiescent lymphocytes are shown here, and they're stained with protein dyes exhibit a very rapid, this is within the first 24 hours, division-independent intensity loss during the first 24 hours. And logarithmically growing U937 cultures exhibit a biphasic loss, with the initial rapid phase being division-independent, so it's sort of illustrated here and here. Being division in dilution should therefore not be initiated until after the rapid loss phase is completed. With membrane dyes, there is no early loss phase, and dilution else can be initiated immediately post-staining. Proliferation dye staining is typically several orders of magnitude brighter than antibody staining. Bristol is same with a membrane dye, either a fixed T0, all up here, sample or an unstimulated sample from the same time point as the test sample can be used to prepare single color, compensation, or unmixing controls, since there is minimal division independent loss. For protein dyes, sort of illustrated here, an aliquot of cells taken immediately after staining is not an appropriate single color control unless it's been verified that the instrument dynamic range allows for these samples to be run under the same voltages as the test samples taken at a later time point. For protein dyes, fixation can also cause additional division-independent dye loss. For time course studies, where the samples will be fixed and run in batch mode at the end of the assay, it's therefore good practice to confirm that fixation does not alter the shape of the proliferation profile. Different protein dyes vary in extent of early division independent dye loss, with CFSE showing greater efflux than cell trace violet or cell trace far red. However, all protein dyes that we've encountered so far exhibit this type of biphasic intensity loss in non-proliferating cells. Question: do protein dyes and membrane dyes dilute out at the same rate? To answer this question, Kata Young-Seo, pictured here, stayed logarithmically growing U937 cells separately or sequentially with cell trace violet, CTV, and PKH26 on day 0. at final concentration of 1 × 10⁷ cells per mil and 5 micromolar dye. After overnight culture to allow for early division-independent loss of CTV, triplicate aliquots were harvested daily for each condition, analyzed on a NovaCyte 3000 cytometer for cell concentration and fluorescence intensity in the for cell concentration and fluorescence intensity. Plotting fold dye dilution, relative intensity decrease, that would be here, versus the increase in cell number, gave a perfect 50% correlation. This would be this line here. Would give conclusions from this double dual labeling cells did not affect cell growth rate as indicated by similar dye dilutions. You can see here or in here. CTV diluted out literally with cell number increases, but much more rapidly, as shown here, than those that were staying with the PKH26. Really an interesting experiment. But really a piece of work with having both of those doing the double staining with the protein and lipid dyes. The analysis here of proliferative fraction, which is the percent of cells that have divided at least once during an assay, is probably the most frequent and easiest metric to use to report dye dilution data. It only requires standard cytometric software and a stained but unstimulated control to establish the region where the undivided cells are located. Using the metric to quantify the response of magnetically sorted T cells stained with CFSE and stimulated with anti-CD3 plus CD28, confirmed the expectation that the CD4 positive, CD25 negative, which is this one up here, subset, which are probably the T effectors, had a proliferative fraction of 83%. And that these had proliferated more extensively than the CD4 positive, CD25 positive fraction, which are probably the Tregs, which is only at a proliferative fraction of only 62%. Similarly, a simple quadrant analysis can be used to analyze two-color dye dilution study and quantitative or quantitatively say that CD4 positive peripheral T cells proliferate more strongly to tetanus toxoid than the CD8 cells, right? Adding concanavalin A to the cultures on day 6 drove extensive proliferation of both the CD4 and the CD8 T cells. However, descriptive analysis or simple enumeration can address several other questions important for the development of vaccines or cellular therapies. The ideal proliferation dye would give baseline separation between peaks corresponding to successive daughter generations, making peak modeling software unnecessary. Sadly, even with the relatively tight intensity distributions at T0 and a strong stimulus, that gives a relatively synchronous response. We have yet to see one that behaves like this. Instead, we use modeling curve fitting software, in this example developed by Bruce Bagwell, to extract the additional information encoded in the detailed shape of the dye dilution histogram. Construction of a dye dilution model uses the same critical controls as an enumeration model. First, the stained but unstained control is used to estimate where undivided cells fall within the dilution profile of the stimulated sample, and the width of the Gaussian peaks are used to model the cells. Next, the assumption that the mean intensity decreases approximately twofold with each division is used to estimate the location of each daughter generation in the stimulated sample. The dye negative control is then used to define where highly divided cells begin to overlap with unstained cell autofluorescence, and daughter generations can no longer be modeled. Finally, curve fitting software is used to vary the exact position, height, and width of the Gaussians to sum the number of daughter cells from each generation, colored peaks here, in every channel, and to obtain the best match between the calculated histogram, smooth black line, which you can just sort of make out over in here or maybe in there, smooth black line, and the observed histogram, the unsmooth black line. This slide, which I like to refer to as our jelly bean slide, as you'll see, illustrates how we can use the relative peak areas and a best fit model to calculate the frequency of responder cells present in the staining population and how much that population must have expanded to generate the dilution profile at the end of the assay. The 100 undivided cells, we're saying there's 100 in here, in generation 0 would have been present at the start of the assay. A single responder cell that divided 6 times would have produced the 64 events observed in generation 6, so over in here, in generation 6. A single responder cell that had divided 5 times would produce 32 daughter cells. Therefore, 5 responder cells would have been needed to produce the 160 events observed in the generation of Gaussian 5. Similarly, 3 responder cells that each divided 4 times would have produced the 40 observed events in generation 4, and 1 responder cell that divided twice would have produced the 4 events seen in generation 2. This model calculates that there were 110 cells in the starting population, 10 of which went on to proliferate, giving rise to 276 daughter cells and 376 total cells at the end of the assay. This corresponds to a responder cell frequency of 9.1%, a 3.4-fold increase in or expansion in the population, and a 27.6-fold responder expansion at the end of the assay. Although not an issue with this data set, it's important to remember that events falling in the dye negative region should not be included when modeling dye dilution because they cannot be accurately assigned to a daughter generation. In general, if the most highly divided daughter cells overlap significantly with the brightest, say 2 to 5% of cells in the dye negative control, the number of daughter cell generations in that model should be reduced by 1. Simple metrics like proliferative fraction capture only a small amount of the information encoded in the detailed shape of a dilution profile. Modeling software converts these shape-related details into a rich set of metrics, providing more complete characterization of biological responses. However, caution is required when reading the literature because terminology is not standardized across programs. When reporting modeling, it is essential to specify that the software and version number used. Mario's article titled Interpretation of Cellular Proliferation Data, Avoid the Panglossian, where he was challenged by a friend to use the word Panglossian in a title, gives a good detail description of the differences. Well, the calculations are all the same, but the differences in the terminology. The amount of information contained in the shape of a dilution histogram varies with the donor, cell type, dye, and stimulus used. This means that there is no one-size-fits-all proliferation model, and depending on the experimental question of interest, it may be necessary to try more than one model and compare them using a goodness of fit metric, or reduced chi-square, which has a value of where a perfect value is a value of 1 for a perfect fit. This study done by Drew Bantley at that time in Johnny Moore's lab included peripheral blood mononuclear cells from 6 different donors. 5 and 6 are shown here. Harvested on 7 different days, stained in parallel with 3 different proliferation dyes, and stimulated for 96 hours with anti-CD3 and IL-2. For PKH26 and CFSE, where generational structure was evident, the fitting guidelines discussed in our study U module lesson 3B, which is just going up online, suggested allowing both peak spacing and peak width to vary as the model adjusted peak height to give the best fit between the observed and model data. For CellView Claret, which lacked generational structure, fitting guidelines suggested keeping peak spacing and peak width constant to avoid overfitting. To allow comparison across dyes, a compromise model was selected in which peak spacing was allowed to vary, but peak widths were held constant at the same value as the unstimulated control. This model gave acceptable fits in all cases, despite donor-to-donor variation in proliferation profile shapes. However, if the goal was to compare multiple stimuli across multiple donors using only CFSE or PKH26 as the proliferation dye, the preferred model would have been one that allowed both peak spacing and peak width to variant. However, once chosen, the same model should be used for all the samples in an experimental group. Question: are distinguishable daughter peaks required for the dye dilution analysis? Based on the full data set from the study shown in the previous slide, the answer to this is no. The extent and kinetics of proliferation responses were comparable for all three dyes, both when the data were analyzed by simple enumeration, as reported by proliferative fraction, and when they were analyzed by modeling and curve fitting and reported as precursor frequency. As expected, all three dyes gave greater donor-to-donor variability when using modeling and curve fitting analysis. This is because modeling takes small channel-to-channel intensity variations into account when making generational assignments. In contrast, simple enumeration ignores such variation and only asks whether a cell's given intensity falls below that of the stained but unstained control. A series of studies by my friends at Dartmouth and the Liège in France illustrated the power of using dye dilution and precursor frequency to monitor responses to vaccination. The ultimate goal of this was a therapeutic anti-tumor vaccine. To test the method, we used 3 different assays for precursor frequency among CD8 T cells after vaccination against the seasonal flu. First, the ability to bind antigen, flu peptide, that's up here, as measured by percent of cells able to bind the tetramer. Second, the ability to measure and secrete interferon gamma after in vitro restimulation with the peptide. And third, the ability to proliferate in vitro after restimulation with flu peptide. The data consistently showed that about twice as many cells bound tetramer as those that responded by producing either interferon gamma or proliferating. The cause of this discrepancy became clear when looking at how tetramer binding cells broke down in terms of extended proliferation after restimulation with flu peptide for 6 days, and that's down here in panel D. At the end of the culture period, responding cells that were tetramer positive, defined as cells having gone at least 2 divisions, are 13.3%, greatly outnumbered non-proliferating cells that were tetramer positive, tetramer negative, positive but proliferation negative. However, the precursor frequency, i.e. the calculated percentage of parental populations that went on to respond, were very similar for both the tetramer positive population, suggesting that 50% of tetramer positive cells in the original population did not go on to proliferate. Barth and colleagues at Dartmouth asked whether tumor-specific T cell proliferation responses after autologous anti-tumor vaccine therapy predicted improved recurrence-free survival in individuals with metastatic colon carcinoma. A PKH67 dye dilution assay was used to determine the CD4 and CD8 T cell precursors before and after bilateral inguinal node immunization with an autologous tumor-lyse pulsed dendritic cell vaccine. Typically stained samples with PKH were run, and the results for a patient at a given time point considered positive if the mean value obtained after stimulation with tumor lysate pulse DCs was significantly greater than the mean value obtained after stimulation with unpulsed DCs. 6 individuals showing detectable increase in tumor-specific T cell proliferation responses at 1 week post-vaccination showed substantially improved recurrence-free survival at 5 years compared with those with no increase at 1 week. Another example of an assay that you can do here is looking at the inhibition by regulatory T cells of T effector cells. Regulatory CD4 T cells down-modulated immune responses and are functionally defined by their ability to suppress the proliferation of CD4 T effector cells. Flow cytometric suppression assays in which T effectors are labeled with a proliferating dye have increasingly replaced tritiated thymidine assays, but in the flow assay, it can be difficult to distinguish between dim, highly divided T effectors from unlabeled Tregs, since both populations express similar levels of CD4. Adding a second tracking dye simplifies discrimination between Tregs, here labeled with CellView Claret, and highly proliferated T cells, here labeled with CFSC. labeling conditions for Tregs were verified not to interfere with the ability of Tregs to inhibit T effectors. That's essentially here where we're getting the same proliferative index, whether they were labeled with claret or not. And the use of a second track also allowed assessment of whether increasing numbers of T effector cells in the assay have an effect on T population, which is basically shown here. So really, what we can see here, using a lot of different colors, how we're able to easily identify the T regulatory cells from the T effector cells. And what was particularly interesting in this experiment was, sure, we're getting lots of proliferation, which was inhibited by the Tregs, but there was also some proliferation going on in Tregs. And so, Kathy, I'm going to stop sharing my screen now and allow you to take over. I hope everybody will bear with us as we move into the next version of the talk. Kathy?
All right. So this is, flip your head around and say, okay, Paul just pointed out that you can have cells that do not respond to a stimulus and just sit there and do nothing. And those cells will stay bright. So that property has been used to find rare stem-like or very slowly dividing cells in this particular example in either normal or abnormal mammary epithelium. So normal breast epithelial cells, if you take disaggregate a population of tissue and put them back in limiting dilution culture where they in a non-adherent environment, about 1 in 10,000 will form these little, what you would almost microspheres, about 300 cell balls, that if you take those apart, you about one, you find usually one or a very few that will go on to regenerate another mammosphere. So they're behaving in a stem-like fashion. If before you put them into these culture, non-adherent culture conditions, you label them with a proliferation dye, and then this example was PKH26, then it turns out that only the very brightest, maybe 0.2 to 0.4%, will in fact regenerate these mammospheres and can go on to do it several times. And if you monitor the way that they're, one of the fate determinants, the num stem cell marker behaves, they are actually dividing that marker asymmetrically. When you do the same thing with cells from mammary, mammary human breast cancer, you find more and more of those staying bright but behaving stem-like cells in the tumors of increasing grades as you go on, as you go from grade 1 to grade 3. And those cells, in fact, if you take those cells and put them back in vivo, the grade 3 cells will regenerate tumors in nude mice much more frequently. So another way of using this kind of dye dilution assay is to look for stem-like behavior of various sorts. And if you are interested in that sort of thing, you can find a variety of examples in the literature with different cell types. At this point, we're going to switch gears and talk about a little bit about troubleshooting and where we've run into problems, or we've had other people tell us they run into problems most often. These are not dyes that exhibit saturable staining. It's not, you can't saturate. You have to therefore optimize your staining. And if you just keep adding more and more dye with either protein dyes or membrane dyes, what will happen is the intensity will go up, the staining CVs will go down, and at some point, but it varies by dye, you will see the cells lose integrity. You just kill them And so the way we usually start when optimizing staining is to run a concentration curve at a concentration of cells that matters, that is roughly what you think you'd want to use in your assay, and pick a concentration that gives you an intensity bright enough to follow the cells that you want through several generations of dye dilution, and that doesn't kill the cells. The next thing you want to do is make sure that you know what the critical factors are for the type of dye you're using. And for the protein dyes, it's most often making sure that you have a fresh dye solution, because there are two esters involved in getting that dye into the cell and to bind to the protein. And if either of those hydrolyze, your staining efficiency will go down. Your staining intensity will matter depending on the staining time and the temperature and how much competing binding sites there are. And we discovered one year in one of the annual courses that actually the different protein dyes vary considerably in how much they are affected, their staining efficiency is affected by the presence of what it turned out to be amino acids in the cell culture medium that they were using to do the staining in. So you just know your dye, learn what works. Same thing is true for the membrane dyes. And there the keys are usually making sure you reproduce the cell and the dye concentration, because it's a partitioning reaction. It's driven by both. They are extremely lipophilic. And so the presence of any salts is going to decrease staining efficiency, because the dyes will just huddle with all the tails in the middle and make little micelles, and the staining will be very slow. So in fact, the kits that come from Sigma have this diluent in them that has no physiologic salts, but is isoosmotic, to try and keep the cells happy long enough for the dyes to hop into the membrane. But the fact that it happens so fast means that the labeling your mixing technique is critical. And again, we had people try different things, and even just injecting dye into cells and and then stopping long enough to cap and vortex gave a much more homogeneous distribution, and trying to inject a very small amount of dye into a large volume of cells gave a real mess. So get the mixing technique right, and the reproduce the concentrations is the trick there. You also need to be sure you have the right controls. You always need a negative biological control. So that's your unstimulated but dye positive. You need a negative instrument control, and in fact, negative biological control. So here's your no dye, but it should be stimulated, because the autofluorescence of those cells can change fairly dramatically on stimulation. And you see this actually more usually in the violet dyes, like Celtrace violet, but you can see it some in the green, blue excited dyes as well. And then the last thing you really need is you have to have a positive biological control. Some cells that have been labeled and have been stimulated with something you know they should respond to, as long as you haven't over-labeled them and killed function. You will probably have to invent your own controls as you're going to a new system. The next thing is actually, you need to have a viability dye of some sort in every kind of dye dilution assay, because guess what? Dead cells shed membrane, they lose protein, and they will lose the label. And our assumption is that when we see a decrease in intensity, that means that the cells have divided, which means we've got to exclude other sources of dye loss. So this was an experiment that Kylie ran, where she did stimulated mouse splenocytes with cell that were labeled with CFSE, did her doublet gating, and did it actually very thorough doublet exclusion based on both side scatter and forward scatter area and height, then eliminated the dead cells based on PI and just looked at live cells, did a traditional lymphocyte gate, and then looked for CD3, CD4 positive T cells. And you can see the gating makes a difference. If here was the ungated, and then if we did the full gating, guess what? There's those beautiful peaks. And so often the answer to why don't my peaks look like that is you haven't excluded other sources of dilution. The other thing we asked with this data set, which was sort of interesting, was how much difference does it make if you get rid of the doublets or not? And as you can see on the right, you get a little bit deeper division between the daughter generations if you do the doublet gating, which is the green filled histogram, versus if you just eliminate the dead and don't do the doublet gating. But that's not always true. You just need to check. The other thing you need to remember is don't gate out the blasts, because the cells, as they, here in this case, our division is being monitored by PKH26 on the y-axis, and we're looking at the forward scatter as the cell size, quote unquote. And you can see as they are stimulated, they get larger and larger. So just be aware that you need to have a fairly large scatter gate if you're going to use scatter gating. The other thing we did with this data set, which was sort of interesting, and you can play with it yourself in lesson 3C when that goes up, because we've given you this data set, is that sometimes it's easier. In fact, I usually find it easier to do any singlet gating after I've eliminated looking at anything else that's not of interest. So in this case, rather than starting with the singlet gating, the sequence went start with a viability dye on this axis, and your marker of interest, in this case, CD3, on the x-axis. Then gate that over and look at your CD4s, which are now up here, and then pull the CD3, CD4s down to your singlet gate. And as you can see, that worked rather nicely, and it makes it a whole lot easier to see where those off-axis singlets are once we've sort of ignored all the other stuff. The other thing that is often an issue for, especially the proliferation dyes, because they're so bright, and we want them to be bright so that we can follow as many generations as we need to pull out those rare responding cells and find them, is color overlap. And what I learned from Kylie on this one was these combinatorial plots are great. You just plot everything against everything. And when you plot it on a bi-exponential axis that's linear near zero and then logarithmic on either side, when you have inappropriate degrees of color correction, you get this backwards kind of tilt. And in this case, the backwards tilt was telling us that the proliferation dye on the y-axis, which was E4670, was being inappropriately corrected in the CD4 channel, which was in the APCH7 channel. And with the original manually set compensation settings, we got this kind of proliferation profile, and that translated to a precursor frequency of 100%. But what was happening was that down here, I'm sorry, the brighter they were on the proliferation dye, the dimmer they were on CD4. So they were getting pulled out of the gate we were looking at, for which was the CD3, CD4. When that was more properly compensated and you didn't have this backwards tilt, all of a sudden, you can see it made a huge difference to the proliferation profile. Okay, we lost cells here. So it is worth worrying about, and it is important to worry about your compensation. You also need to worry about your cross laser excitation, and this is actually seen very nicely in the CYTO Aurora data that Monica kindly collected for us. These are two far red dyes. The cell trace far red is a protein dye. The cell view claret is a membrane dye. And both of them give signal, cross laser signal. Their quote unquote primary channels would be out here in the far red, but they both give signal and, excited by all the other lasers at concentrations that you might want to use for a proliferation dye dilution experiment. And in this case, it also turned out that the cell trace far red gave a little more signal than the cell view claret in the violet excited channels, measured where you were measuring the red emissions. So if you had a critical violet marker that you needed, you might make a different choice based on knowing what these profiles are like. So again, it's a matter of know your dye. Other lessons learned the hard way? Well, don't try to fit daughter generations down there where it's overlapping with your stimulated but dye negative control. If you don't collect enough cells in each part of the distribution, you're modeling noise, and that will give you either unreliable results or incorrect results. You do need to worry about not compensating. You need to worry about correcting for or eliminating division unrelated causes of dye dilution, dye loss. And you need both positive and negative controls to be sure that you're not compromising cell function with the way you're labeling. And the usual problem is indeed interference from the tracking dye and the channels used for other probes, but it can be a problem the other way too, when you have a highly divided cell and a relatively bright protein or antigen marker. Just a quick recap. What are the differences between the two dye types? They're mostly in how you optimize and when you start your assay. But after that, the rest of it is about the same. You need to be sure you haven't overlabeled. You need to be sure that you're able to detect your other markers in the presence of the bright undivided cells or less divided cells. You need to be sure you're eliminating other possible dye loss reasons, and then you need to decide how you want to analyze your data. Paul gave you several different ways. Jumping back up a level, so think about DNA content measurements of dot proliferation versus dye dilution radiated. Here are the big things that can go wrong. If you have, and the consequences are different. If you're staining for DNA and you don't have enough dye, your CVs will go up, you'll get a low G2-G1 ratio, your percent S will be too low, and you're likely to miss your diploid populations. If you have not enough dye per cell for a dye dilution assay, you'll also get a higher CV, and you'll get less intense staining, and you will be able to resolve fewer generations. Doublets and aggregates are a problem for both types of proliferation assays. And dead or apoptotic cells are also a problem for both types of assays. So basically what I'm telling you is I'm making a big plug for going, if you want to get into any of these details, just go back and sign into CYTO and look at lesson 2 or lesson 3. So what have we learned? Well, at least since I started doing proliferation assays, we've got a lot more markers and a lot more ways of asking the questions and answering the questions. And the answer is, it depends. We just have to know on what. And by sharing what we've each learned the hard way, we learn more faster. So with thanks to all of our collaborators who've educated us through the years, it's your turn to ask questions. And I will stop sharing.
Thank you so much, you two. So like Kathy said, if you've got any questions, please just pop it into the Q&A box. There's nothing in there at the moment. So many useful tips and tricks. And if anyone's done proliferation assays before, I'm sure you wish you knew this stuff before because it has, some of it's been learned the hard way. So I've got a question just to start us off. This is something I've been asked before. Paul, you kind of touched on it. So it's about a Treg assay. So the question is, I labeled cells in a Treg assay. The effector T cells were labeled with CFSE, and the Treg cells were labeled with cell trace violet, then stimulated with anti-CD3 and IL-2. Many times I see the Tregs dividing too. Aren't they supposed to be inhibitory? And why are they dividing?
That really is an interesting question, and I'll share my screen. I can. So that's basically this experiment here, if I understand correctly. This was a great experiment that Joe did, and basically the idea was that we really want to know is if we increase the number of T regulatory cells, which is going along this axis. This is the did we decrease the amount of proliferation? You can see where there's a low concentration of Tregs. There's a fair amount of proliferation. But as the concentration increases, where here's 2 to 1 Tregs to effectors, the proliferation decreases. And what you can see, I think, to your question, is that there is actually some proliferation. The cell view claret was the one used for the Tregs. The CFSE was the one used for the effector cells. That there actually is some proliferation, and that, frankly, was a surprise to us. And as I recall, the amount of proliferation that we saw decreased with the more Tregs that were in the assay. So it appeared to be sort of a function of how many, what the effector to Treg ratio was. I honestly think what's going on is that these guys, they're proliferating away. They're having a great time, and they're producing lots of cytokines. IL-2 would be the one that I would think of, but probably IL-15, IL-7, and a variety of other ones. But you can see here that they're producing all that, and that's having a bystander effect on the Tregs. That's what I think is going on. But it is clear that the more effector cells that we had in here to the fewer Tregs, the more of the proliferation we saw in that population. So yeah, we expected that these cells would just be quiescent and they wouldn't do much, but it turned out that there was some proliferation. Hope that answers the question. But this is a beautiful experiment that Joe did, and you can see he's doing all the things that Kathy told you, probably because Kathy told him how to do the experiment. But where he's gating out the dead cells, he's doing a nice general forward versus side scatter, and then we're using the CD4 markers as a way of gating in on the cells.
Yeah,
how's that?
Excellent. That's great. Thanks for that. Nice work, Joe, as well. I've got another question here. So it says, is there a dye that maximizes the number of divisions that can be seen, or an instrument that has shown the maximum number of traceable divisions?
Boy, is there a winner of everything? No, I don't think so. I would say that the fact that we now have instruments that have, second decade, 7 decade scales lets us go wild. It really does depend on the cell type and the dye. You would think that a far red dye would be the best in some ways because there's less autofluorescence. And so you should be able to march down scale farther before you bump into that low end where you can't tell how many divisions there are. I haven't always seen that, but it is somewhat instrument dependent. And I would say, as long as you don't have other critical markers that mean you have to back off on your proliferation dye staining intensity in order to avoid being unable to do the necessary spectral overlap corrections. If that's not an issue, you can usually see at least 6 and maybe 8. I have rarely seen anybody claim 10 or 12. And I'm not sure I believe it because in some cases I think they were probably fitting either noise or that region, which was where daughter generations can no longer be distinguished from unlabeled cells that were present in the assay.
Yeah.
Yeah, I would just say that I think my favorites, but this is would be the cell trace violet if you're talking about a protein, and the PKH26 if you're talking about the lipophilics. But that's just I haven't done any scientific. I wondered, Kylie, if you looked at instruments. Have you ever compared any instruments?
I haven't gone down so much down the instrument path. Cell trace violet and PKH26 are my faves.
It surprises me about because I would have said cell trace violet too, but that it has the biggest bump up in the autofluorescence too. So there's actually a very nice paper by Parrish, or by Qua and Parrish and some other colleagues, where they compared a bunch of the protein dyes on the how much the autofluorescence changed, and it turned out honestly that they were all about the same by the time you accounted for the increase in terms of their dynamic range for being able to tell divided cells apart from autofluorescent cells. They were all about the same, except maybe in the red. It just kind of, it looked like you should be able to go farther down, but you couldn't because the floor was higher.
Yeah, that makes sense. Well, thank you guys so much. I want to thank everybody for attending the webinar today. I hope you got a lot out of it and learned a lot. Oh, hang on. There's one more question. Better to get this one. Svetlana, does autofluorescence extraction using the full spectrum instruments help with the resolution of tracing dye's resolution?
I would think it should. I have not personally done that, and it's on my agenda.
Yeah, there we go. I would think it should too. Yeah, so I just want to thank everyone for attending today, and it's been a real pleasure connecting with you all. If you're interested in learning more about proliferation, because it's such a fascinating topic, then please be sure to check out the latest module in the Proliferation by Flow Cytometry series on CYTO U. Our latest modules just dropped, so that's exciting. Also be sure to sign up for the next webinar on October 22nd. This is the first in our data analysis series, and I'm really looking forward to that. At the end of today, you'll receive a short survey. If you could take a moment to fill that out, that helps us deliver the quality and the material that you guys are interested in. And also, if you want to re-watch this webinar, it's been recorded, it'll be out soon, and you can watch it at your convenience in the future. Just want to thank everyone again, Kathy and Paul, for your time. I want to thank ISAC and the Education Committee, and Anna Sarnowski, the Director of Programs, for all the work she does behind the scenes. So thank you all and have a wonderful day.
Bye now.